everybody. So if you're watching this video, that probably means that you have looked over the acids and bases notes, or you were here, and now you're ready to go ahead and start testing some of them. So it's really important that we know the difference between acids and bases, right? Acids have a pH of 1 to about 6 and have a hydrogen ion in them. That is what makes them an acid. Whereas bases have a pH level of about, let's say, 8 to 14 and have hydroxide ions. That's what typically characterizes those compounds. Now, in order to know if something is an acid or a base, right, if we have a random liquid that we find somewhere, we may want to test to find out which one it is. And you can do that using a lot of different indicators. In the notes, we talked about, I want to say about 4 or 5 different ones, and we will be testing them today to see how they work in real life. So we have blue and red litmus paper here. We have universal indicator. We have bromomethyl blue, phenolphthalein, and cabbage juice. It's kind of a funny one, but there are actually natural acid-base indicators that you come across every day, well, sometimes, in your everyday lives. So cabbage juice is one of them. Another one is poinsettia leaves, or even hydrangea flowers. So based on the color that they turn, you can tell if the soil is acidic or if it's basic. So today we'll be using these 5 different acids and bases to test out some chemicals that you come into contact with almost every day. Okay? So hopefully you guys are using some of this right now. Some Clorox spray, some type of disinfectant. We got vinegar, vinegar, lemon juice, laundry detergent, tap water, and some salt water. So we're going to be testing today to see what those chemicals are. Are they acids, bases? Maybe take a quick moment right now just to think back in your head and say, what do we think? Do you think vinegar is an acid, a base, or neutral? There's 3 options here. So I'm going to go ahead and get us started on this lab. So feel free to follow along. All right, so to do this lab, you are going to need this handout right here. You can either print it off on Google Classroom and follow along with us that way, or you can also digitally complete it online as you are watching this video. Okay, so let's follow along and see what we need to get. We need some gloves and goggles. Got my gloves, got my goggles on as well. Very important, especially if you're testing acids and bases and you don't know how strong they are. You definitely want to make sure that your hands and your eyes are protected because acids and bases can be corrosive and you want to make sure that you're protecting your skin and your eyes. All right, next up we need a well plate map and a well plate. I have those right here beside me, as well as our indicator bottles, which I just showed you. So we're going to be testing bromomethyl blue, phenolphthalein, universal indicator, and cabbage juice, as well as your red and blue litmus paper. We're not going to worry about the pH paper for today, as well as 6 household substances, which I just showed you a moment ago. So what we are going to be doing is using our well template and my well plates. They fit nicely over top. And I'm going to get a small sample of 3 of my unknown acids or bases. So we're just going to pick, let's pick for now, tap water, laundry detergent, and some lemon juice. Okay. So in order for me to remember this, right, I'm just going to put a small note here saying that A is tap water, B is laundry detergent, and C is lemon juice. Okay, so I'm going to go ahead and put 5 drops using my pipette of each substance in each row of my well plate. All right, so now that that is done, it's time to add our indicators. I'm going to put my samples over here for a minute. All right. So in my first well, I'm going to be testing my blue and red litmus paper. Second well is going to be my universal indicator. Number 3 will be bromomethyl blue. Number 4 is phenolphthalein. There we go, that's better. And our 5th category will be our cabbage juice. So I'm going to go ahead and write those down at the top here so that I can keep track of that as well. I don't want to, forget. So litmus paper,
universal indicator,
awesome. All right, so first thing we're going to do is start with my litmus paper. Okay. So I'm going to grab a piece of some of my red litmus paper and my blue litmus paper. Now, red litmus paper, the trick to learning about litmus paper and whether it's an acid or a base is that whenever it is an acid, your litmus paper will turn red. If it's a base, your litmus paper will turn blue. And if you put both colors in and neither of them change, you know that it is neutral. So we're going to go ahead and test each of these right now. Okay, so I have my red litmus paper. And while I do this, it's important that you record your results in your lab. Okay, so right here, right, we should be writing what substance I am testing. So we are going to be testing tap water. Let's go ahead and write that in. Sorry for the messy writing, guys. Tap water. And I want you to write down what color it turns for my red litmus paper, my blue litmus paper, and then we're going to continue on with the other types as well. Okay? So, my red litmus paper, go ahead and dip it in. There you go. Okay. So my red litmus paper stayed red. My blue litmus paper stayed blue. Interesting. Okay. Next up, we are going to test out the laundry detergent as well. Okay. Red litmus paper. Whoa. So that turned blue. And it remained blue. Oh. And for lemon juice, it turned red with my blue litmus paper and it stayed red with my red litmus paper. Okay. All right, so now that you have those tests, those columns should be complete for you. And And now we're going to try our universal indicator. Just need 2 drops in each well. You can see those are some very different colors. Very interesting. So, what we can use, you might be thinking, what do those colors actually mean? Oh. Sorry guys, class is still in. Okay, so because you might be wondering what those colors mean. And this is where this comes in. So based on the color and the type of indicator, you can figure out if it's an acid or if it's a base. Now, some of these indicators that we're going to be using are very specific. They're going to tell you if it's an acid, if it's a base, if it's neutral, and how strong it is. Other ones are not as specific. They're just going to tell you if it's an acid or if it's a base. Okay? So our universal indicator here has lots of different options. And when you color match it, we can actually figure out what the pH is and how that means how strong of an acid or a base it is. So all you have to do is go ahead and color match. I'll help you with the tap water and you guys can do the remaining ones. Okay, so we're going to color match my tap water. I'd say that it's either between lime green or green. Okay, so that means that this either has a pH of 6 or 7. So I'm going to say it's green. That's my guess. Okay, so it probably has a pH of 7. That is what that indicator is telling me. Okay, so let's fill in some of this together. So red litmus paper stays red. Blue litmus paper stays blue. We haven't done the bromomethyl blue yet, sorry. We're on universal indicator, which is green. Okay, so this wants me to record the color and any pH number associated with that, okay, which we found out was 7 based on the picture. You don't need to include that for these two. So now let's go ahead and test out bromomethyl blue. Okay. Give it a little swirl. All right. So with bromomethyl blue, I have 2 different colors. They kind of have a yellow and a blue. And that's where you're going to now use your bromethymol, blue pH indicator. And you can see if it's yellow, it's somewhere in this range. It's somewhere with a pH of 0 to about 5.5. which is acidic. If it's basic, it's going to be about, I'd say about 9 to 14. And if it's neutral, it's going to be some shade of green or slightly acidic or slightly basic. So if I look at here, this one is yellow. Sorry, the blue's kind of reflecting off of it, but it is yellow. Okay, so that would mean that it's acidic. It's some type of acidity level from 0 to about 5.5. My laundry detergent, well, we know what that it's a base. We just don't know how strong it is. It could be, 9. It could be very strong. It could be 14. Okay. And same with lemon juice as the tap water. So I'm going to record my results for the water, my tap water. It's yellow. And let's put the pH range. So it's probably about 1 to 6. Okay. So you should be following along. At this point, you should have your 2 other substances filled in, your laundry detergent and your lemon juice. You should have your red and blue litmus paper recorded, your universal indicator recorded, and now your bromomethyl blue indicated. Okay. Now we're going to move on to phenolphthalein. So phenolphthalein, when I look at my indicator chart, tells me some things. If it's alkaline, which is another word for basic, it's going to be this bright magenta pink color. If it's neutral, it's going to be white or clear. And if it's acidic, it's also going to be white or clear. Okay? Boop. Okay. So now I can go ahead and record that information in here. Okay. So tap water with phenolphthalein is clear. So that has, when I look here at my chart, somewhere between, sorry, this is 1 to 6. We know that neutral is 7. So it's somewhere between the range of 1 to 7. Okay. Go ahead now and fill in your laundry detergent and your lemon juice. You have your information right here as to what color and what pH it is. And the last one we're going to do is our cabbage juice. So I've got my cabbage juice right here. And cabbage juice is kind of like universal indicator. It gives you a color for almost every pH level, right? Universal indicator has the most color options. It gives you the most accurate results. You not only know if it's an acid, a base, or neutral, acid, a base, or neutral, but you also know, based on the color, what the specific pH is, which can be really handy. Your cabbage juice also does that. It also gives you a nice color range. It can tell you if it's very basic or very acidic. However, it doesn't have as many colors, right? It goes up almost in groups of 2. So I can kind of tell my pH, but it's not going to be as specific as this one. It's still very helpful though. Okay, so we're going to go ahead and put 2 drops into my tap water, laundry detergent, and maybe a few more, and lemon juice. There you go. Give it a little stir. Looks like a rainbow over here now. It's awesome. Okay, and so now I can go ahead and compare these guys right here. Okay, so I have my water, which is kind of, I'd say, I don't know, it's a darker purple, or blue, I mean. So I'm going to say it's about indigo. Somewhere between purple and indigo. Okay. Next one, it's kind of a green color, right? So the only green we have is about 12. And my last one, my lemon juice, is that pinky kind of color right here. So it's somewhere between the red and the pink. You tell me what you are observing right now in your chart. Okay, but right now we are going to fill in just the tap water. Okay, so I observed somewhere between a pH, it was kind of purple to blue, so its pH is between 6 to 8. Okay. Somewhere between purple to indigo, sorry, I should correct that. And so it's somewhere between 8 and 6. ahead and add in your information for your other two samples. You should now have this entire portion filled in as well. All right, so now I'm going to dump this out. I'm going to rinse it and dry it really well. We don't want to have any residue left in our well plates because we are now going to try our next three samples and we do not want them to be contaminated. All right, so I'll be right
welcome back.
Okay, so now we are going to be doing our three other samples, which are salt water, Clorox spray, and vinegar. All right, make your predictions now, folks. All right, and this time I'll make sure I do it in the order that it is on your chart. So we'll do litmus paper. Sorry.
Okay, guys. Blue knuckle blue. Phenolphthalein. Universal indicator. And cabbage juice.
All right, so let's get started. Okay, 5 drops again of my samples. All right, so now that we're all set up and we have our samples in our wells again, we're going to go ahead and try out that litmus paper once again. Okay. There we go. So okay, so our red litmus paper stayed red. Our blue litmus paper turned blue, or stayed blue for salt water. So make sure you're filling in that chart. Our Clorox spray, oh, it's too blue, oops. Just seeing if you're paying attention. So our red litmus paper turned blue. Our blue litmus paper stayed blue. All right, so vinegar, red litmus paper stayed red. And blue litmus paper turned red. Next up is our blue indicator. Okay, and just a nice reminder, right? Pause the video right now if you would like to remember that an acid is going to be yellow. It has a pH of around 0 to 5.5. Bases are blue with pH of 14 to anywhere to about 9. And then anything neutral or slightly basic or slightly acidic is green. Okay, so that is the information that you're going to need for these samples. Okay, so make sure that you write down if it is the color and the possible pH range. Okay. Once you're finished that, we are going to move on with our phenolphthalein. Also a kind reminder, right? You can pause the video now. Phenolphthalein is going to be bright pink with pH range of about 8 to 14. It's going to be basic. And acidic from about 1 to 7. So that's acidic to neutral. All right. So based on these colors and what your indicators are telling you, you can tell that this is either an acid or a base. However, none of the indicators yet have told us how strong of an acid or how strong of a base it is. That is definitely what our last two, our universal indicator and our cabbage juice, are going to help us to do. All right, so our next one is going to be the universal indicator. All right. It's getting colorful. Okay, so this is where you'll use this chart. Okay, so you're going to try and match it up with the color that best suits it. And then you're going to tell me, okay, what's the number that matches it as well? So tell me the color and tell me the pH number. All right. And our last one is cabbage juice. Ooh. All right, so this is now where we are going to use this indicator sheet. Okay, so you'll know, right, which, if it's an acid, if it's a base, and you have an idea of how strong or how weak that acid or base is based on the color that it turns on this chart. Awesome. All right, so once we're finished that, I'm going to clean this up very quickly and I'll be right back to walk you through those questions that are on the next page. All right, so you should have this chart and it should be full of all of your information now. You should have your 3 other samples listed and what color and what pH number is you observed from our lab that we just did together. On the next page, you have some data analysis. So, based on all of your testing, fill in the 2 charts below. Be specific with the color or color ranges that appeared with the two different acids or bases you tested. Well, we tested more than that. What I actually want you to do for this is use this piece of paper that you should have also printed off as well. If not, that's fine. You can use the one in the video and just pause it so you can get a better look at it. I've done an example for you. So for each of these indicators, I just want you to tell me what color or color range does it turn when it's in the presence of an acid or when it's in the presence of a base. So you know that red litmus paper is going to stay red in the presence of an acid, and it's going to turn blue in the presence of a base, right? Blue means base, red means acid in the terms of red litmus paper, or blue litmus paper as well. For bromomethyl blue, you're going to want to look here and find that indicator scale and tell me what color does it turn when it's an acid versus what color does it turn as a base. Same thing for your phenolphthalein. And for your universal indicator and your cabbage juice, please tell me the color range. So just tell me, a range of colors that you could find when you test an acid versus when you test a base, and fill in the chart. The next table is asking you to list the household samples that we did together, write the pH value that you think it is, and whether or not it's an acid or a base, based on the colors that it turned and your knowledge of what that means. Okay. Last little bit is some questions. So, can either red or blue litmus paper be used to identify acids? Do you have to use red? Do you have to use blue? Or can you use both? Explain. So, number 2, during the lab, you used a variety of indicators which told you if the liquid was an acid or a base. Tell me which one was the most accurate, which one gave you the most data, and which one was the least accurate. Which one maybe only told you if it was an acid or a base, but it couldn't tell you if it's neutral, couldn't tell you how strong it was. Okay, and explain your thinking. Which one is which? All right, list the general properties of acids and bases that you have learned / discovered during this lab. I also want you to use your notes for this. So you should have watched or listened or read the note that was posted, or you were here the day that we went through it. And I just want you to pick 3 points to put down that identify acids. It could be how they taste, whether or not they conduct electricity, maybe what pH range it has, and do the same to compare them. So compare your acids and bases on those characteristics. And the last question is just comparing your household substances. Which ones were acidic, almost neutral, or basic? All right, and that's all. So I hope you enjoyed this lab. Hope that you're, having a good time inside and staying safe. Feel free to email me a picture of your completed lab pages, or you could also use the Dropbox that I'm going to put on Google Classroom. All right, have a great day, everybody. Miss you.